Okay, so to get started, I am going to give you a little overview of Imaris. I'm going to be using an application called Prezi. I hope no one gets motion sickness, but we'll see. And then right towards the end of the session, I'm going to jump into the software and just give you a little tour of where things are and how the workflow, or how to use Imaris and build a workflow for analysis. So to get started, if Prezi wants to agree with me here. There we go. To get started, I want to focus on 4 main areas. I'm going to go through some acquisition considerations. So when you acquire your data on a microscope system, things to bear in mind if you're going to be bringing it in to do some 3D visualization. I'm going to go through different visualization techniques, so how we render the data, how you can optimize that visualization, and also how to export some of that data as well. I'm going to focus mainly on our analysis options because I believe that's probably why you're in the room today. So analysis is done mainly with modeling, so I'm going to spend some time talking to you about computerized modeling. And then lastly, I'm going to go over a little bit of data interpretation. So Imaris is going to give you the ability to collect a huge amount of data. And so I want to show you some new tools that we have to let you kind of look at trends and similarities and differences between different data sets. Okay, so we're going to jump right into acquisition, and I'm going to start really simple, so I apologize for starting this basic, but basically when you collect data on a microscope system, you are taking a series of 2-dimensional slices. We take those slices and we use the metadata associated with the files, and we bring that all together into a volume or a cube of data. What was previously a 2-dimensional pixel becomes a 3-dimensional voxel of information, okay? The reason I highlighted metadata is it's an important component of what we need in Imaris in order for you to get data and get your visualization out. So I'm going to jump down to file format support. So as far as data acquisition, we support 50 different file formats. In fact, just over, I think it's 51 or 52. The reason we spend so much time working on file readers is so that you can keep your data in the proprietary file format that you acquired it in, bring it into Imaris, and we know exactly how many slices you have. We know what your pixel resolution is. We know what magnification, how many channels, the time points, all of that good stuff. So metadata is basically what tells us how the file is organized, okay? So I highlighted the big 4 microscope companies up top. They each have multiple different file formats, which are all supported in Imaris. So I highly recommend if you are acquiring from one of these instruments, keep it in their native file format and bring that into Imaris. It's going to save you a huge amount of time, okay? We also support a lot of other, maybe not as well-known formats. I've got Slidebook in there, Metamorph, LaVision. Now this is a unique one, LaVision Biotech. They make light sheet microscopes. They actually export in our own file format, so those files come straight into Imaris. I have Applied Precision, so Delta Vision System, OMX. I have Gatan here, PerkinElmer. So I have a number of different file formats that can come straight into Imaris. One that's missing on this list is a TIFF file. So I want to talk to you about TIFF files. They work perfectly fine. You can import them, no problem. Everyone uses them. They're universally supported. You can open them anywhere you want. One big issue with a conventional TIFF file is it does not store metadata. It has no information for us to be able to pull out what the step size was, what the pixel resolution is, all of the things that we use to build that 3D volume. So TIFF files in general will require you to enter that information in by yourself. So if you have an option and you can use one of our supported formats, so maybe you're using a Zeiss or a Leica system, keep it in their format rather than exporting out as TIFF. Or if you're using TIFF, also export out the LIF so you can bring that into Imaris. Okay? Now, there are some special TIFFs out there. And there's a TIFF format that Micromanager spits out. There are TIFF formats that the Prairie system spits out. They typically have little XML files with them, which we read. That is not the case for all XMLs. So if you're in a situation where you have a system that's generating TIFF files, come talk to me. We can always set up something where we can maybe have the software read your XML. So let me know about those situations. But for the most part, TIFF files, if you don't have the metadata, it's going to be a little bit harder for you. Okay, so I want to talk a little bit about imaging modalities. It used to be back in the day that people associated Imaris with confocal imaging. Sorry, I keep blinding myself with the projector. So nowadays, it's a lot more. There's a lot more instrumentation available, and a lot of people are using different modalities to generate volumetric data. So other than confocal, a lot of people using light sheet, a lot of people using multiphoton, spinning disks, super res. We're also now seeing a lot more people using micro CT. So if that is something that you're interested in, ultimately anything that generates volumetric data, slices of data, you will be able to bring into Imaris for visualization. Okay? I have a tiny little image at the bottom of an electron microscope image. Those are also supported, so you can also bring electron microscopy data into Imaris. Okay. Now, anyone in the room generating files larger than about 1 gigabyte in size? A few. Okay. For those people who are in the gigabyte plus range, and if you're not there right now, you're probably going to get there soon. I'm bringing this up because the bigger your data, the harder it is to manage. It's harder to transfer, it's harder to process, it's harder to visualize. So we've spent quite a bit of time making sure that our file format is optimized and that Imaris is optimized to handle big data. So when you start generating time-lapse data, or you're using a confocal and you're tiling, you're taking different fields and stitching them all together, or if you're using a light sheet instrument, all of these data sets are going to start getting bigger and bigger as your experiments and your instrumentation, changes. So Imaris has a multi-resolution image pyramid that we use as our file format. It allows us to interactively change the resolution of the visualization based on how you are zoomed in or zoomed out. So I have a little illustration here. It's a kidney section, I believe. So I'm going to zoom up and let you take a look at this. Now, the colored blocks on here are indicating different resolution levels. And as you zoom into that kidney, you'll see that you're going to see more and more blocks. And the more blocks indicate that we're looking at a higher resolution. As you zoom out, you're starting to see less of the details, so the blocks get bigger. We're using a lower resolution visualization or rendering. So this is happening interactively when you are using Imaris. You're not going to notice any of that resolution change. Is happening dynamically as you are playing with the software. It's kind of, if you imagine Google Earth, or if you're looking at Google Maps and you look at the world and then you zoom in on San Francisco and you can see all the streets and buildings and things like that. It's the same concept. We use the resolution size that is appropriate for the detail that you can see at that zoom level. Now, the benefit of using a technique like this for reconstruction is that you can have a system with very little RAM and open up a data that is significantly larger than the amount of RAM you have on your computer. So this is something that with a laptop, you can actually open up a very large data set purely based on the file format that we incorporate. Now, for those people who are in big data world, we have a free application that anyone can download and install in their computer. It's called the Imaris File Converter. It runs on Mac and PC, and it allows you to convert your files into Imaris files, and that's all it does. It just does this conversion from one format into another. The reason that is a benefit is it will cut down the length of time it takes to open and visualize your data when you're in Imaris. If you open up the original file, it can take a certain amount, like quite a length of time depending on how big the file is, in order to see it and be able to work with it. If it's converted into the Imaris file format first and you open that, it's going to open up instantaneously. Okay? So just to give you an idea, I'm showing you some boring graphs right now. Over here on the left-hand side, we opened up a TIFF series. It was a terabyte TIFF series. We opened that directly in Imaris, and it took us just under 4 hours to open and visualize and be able to start doing anything with the data. So that's us going into File, Open, and then to try and open these TIFF files. If that had been converted in the file converter first, it opens up in 25 seconds. Okay, so 4 hours or 25 seconds. I think it makes sense to convert it first. Now, the file converter is batchable. It is multi-threaded, so you can have it running in the background and doing the conversion for you. So it could be running on your acquisition system, it could be running on your laptop, but this little conversion process doesn't take a huge amount of time, or it's not a huge amount of processing either. So it's something I highly recommend if you guys are going to be working with a gigabyte plus. Okay. Incidentally, the Imaris file converter, if you go onto our website and do a search for it, it will give you the link where you can download a license for yourself. Okay. Next thing, I want to talk about 2 new features that have been recently added, which give you the ability to do something that for existing Imaris users you were not able to do previously. So the first thing is we now have a multi-image visualization. So what that means is previously you would open up one data set and you would visualize and render and analyze that one data set. Now you can open up to 16 different images of different sizes and different resolutions in the same 3D space. So I have an example here of 2 different modalities. I have an EM image and a confocal image that can be brought into the same 3D space and aligned, and then you can make measurements on each of those images. Okay? Now this is just one example. Another example would be if you're imaging a whole mouse brain and you cut it in half and you image those 2 halves, and now you've got 2 data sets of 2 halves of a brain and you want to put them back together. This multi-image viewer will allow you to bring them into the same 3D space and put them back together as one organ. Okay? Another example would be if you're using different resolutions. So let's imagine you're looking at a Drosophila embryo and you want to be able to see the whole embryo, but you want to zoom up and focus on a particular region at a higher resolution. You can now take a lower magnification image, import that into Imaris, and then add in a high resolution image and have it aligned in the correct location so that when you zoom in with the renderer, you can see a high resolution reconstruction. Okay? So this one, I don't think it was given enough marketing attention because I use it all the time. I even use it when I'm comparing images that have been processed versus not processed, so deconvolved versus non-deconvolved, or if you've applied some kind of processing to it. It's nice to be able to import multiple images. Okay, so just really quick, once you have imported your different images, you can actually go through currently a manual alignment process. If we read stage coordinates, we will align them based on the stage coordinate information. So that is currently the case for Leica LIF, Zeiss CZI, and Delta Vision files. So those 3 currently we read the stage coordinates and we'll put them based on the stage coordinates. But for multiple different modalities, you would have to bring that data in and manually adjust or align those 2 together. Okay. The second new feature is the Imaris Stitcher, specifically for people who are tiling, who are taking multiple images at different locations and want to bring that data all back into one file for visualization. So a lot of acquisition platforms provide a tool like this. So if you can tile the data, you can typically stitch it in that platform. We've actually come up with a solution because there seem to be limitations to the acquisition system's ability to handle bigger data. So this has been tested all the way up to 2 terabytes in size. So if you're working with lots of tiles, or if you're working with a few very big tiles, you can use the Imaris Stitcher to stitch those together. Okay, so just a little illustration here. So this image, I believe in total is just under 40 gigabytes from a light sheet. We're looking at green vasculature, not sure what the red is. Basically, the little squares that you can see on the screen are representing the different tiles. And what we do with Stitcher is we bring them in and we align them either based on a grid format that you enter or the stage coordinates, and then we align the data based on a channel of interest, and only the data within the overlap region is used for the purpose of alignment. So if I zoom in, I'm just going to show you this zoomed up. So here's one of the blood vessels. You can see these overlap regions in this kind of narrow part of the image. And you'll see that the blood vessels are not aligned properly. So this is very common when you do tiling. You'll see that they're not perfectly aligned. So what the stitcher does is it looks for in those overlap regions, and it will use our stitching algorithm to align those images. Now, in some cases, alignment is not necessary, so the alignment part is your choice. You can just bring it in and just have Imaris stitch it based on its current coordinates. But in this case, you can align it first and then output it as one whole image. I'm also just going to show you, so this is now that whole tiled file reconstructed and visualized in Imaris. Okay? So 2 new features to basically allow those people who are working with different modalities, different resolutions, different tiles, a way to import that data. Okay, so we're going to move on to visualization. Anyone feeling a little motion sickness yet? Okay, so we're going to zoom in now to visualization. So in this component, I want to go over a couple of different rendering techniques that we provide in the software. And it's actually surprising to me that most people who have used Imaris for years don't actually know that there are other rendering options available. We make it a little difficult for you to find them on your own, so I will show you that in the tutorial sessions this afternoon. So I want to go through these different rendering techniques and why we have them. And then I'm going to go through some image processing and export options with you as well. So first and foremost, I have an awesome mitotic cell rotating in front of you right now using a maximum intensity projection, which is our default rendering technique and probably what most of you are familiar with. The 3D or the maximum intensity projection in Imaris is a 3D maximum intensity projection, meaning that whenever you grab it and rotate it, it will automatically change based on the orientation of the data. One of the limitations of a rendering technique like a MIP is that there's no depth information. So if you are taking a snapshot of this image to put into a publication or a presentation or a poster, it's very hard for anyone to actually tell whether that's a slice or a 3D reconstruction. So the reason we have other rendering techniques is to give you that illusion of 3D on a 2-dimensional platform, screen, a monitor, a piece of paper, okay? So the next one I want to point out, and for some reason it's really dark, it's a lot brighter on my screen, you'll just have to take my word for it. This is called a blend. It's also referred to as an alpha blend. We did not invent it, but basically this technique does a couple things. It adds opacity to the data set. That opacity is based on the intensity value of the voxel. We also apply shading, and those two things together give you the illusion that it's no longer a flat structure, but that it has some 3-dimensionality, okay? So we also turn the background gray. The reason that we do that in Imaris is to help to emphasize the shading and the opacity, okay? So opacity is adjustable, so you can make it more or less opaque based on whatever it is you're looking for, and each channel can be adjusted independently of one another, okay? So the nice thing with this is I can now see that those spindles were all the way around the chromosomes. I have now some spatial idea of what's going on with that structure. Now, the next technique is called normal shading. Now, normal shading is pretty much identical to the blend with one difference. We add an additional light source. So it's like we have a flashlight that you can grab and orient in whatever direction you want. And it allows us to cast basically a gradient of light to help emphasize and put focus on a particular part of the data. So in adding that additional light source, so this little yellow arrow I have here is showing you the orientation of the light source. That is adjustable by yourself. But you can move that flashlight and have it shine or direct light in a particular orientation. It also really helps to emphasize surface structure. So if you're looking at muscle cells, if you're looking at the outside of an embryo, you can really highlight the surface structure with this technique. Okay, we now come to my favorite, also the most computationally intense, rendering technique. It's called a shadow projection. It's also called, internally, we call it the money shot. And the reason for this is when people use this, they're typically using it to give you the most three-dimensional looking structure possible. Okay? We're trying to convey 3D as best we can. And this is usually the rendering technique. If someone is lucky to get a journal cover, this is the rendering technique you want to go with. Okay, so what it basically does is the same as the last two techniques. We turn the background gray. That is a preference. You can have it whatever color you would like. We do that to help emphasize the edges of the structure. Instead of having it black, we can kind of see edging and shading. But what we do is that same light source that we just talked about, we still have that light source in place. But what that does, it actually casts shadows behind those structures. So if a structure is obscuring another structure, that one behind it will appear black. So it even more so gives you that illusion that you're looking at a three-dimensional structure. Okay. Now this one will take a little time to render if your data is big. Now if it is 2 gigs or under, you probably won't notice a difference, but as the data set gets bigger, you'll notice it will start to take time. Okay. So if this is what you really want, then be patient. It's worth it. Okay. So this one, we're going to go over these rendering techniques in the application tutorials this afternoon so you can see how to optimize. Okay, so a little bit about exporting. So we've just seen different rendering techniques. We can export the different visualizations. We have galleries and section views and slice views. The most common way to get data out of Imaris for presentation purposes is a movie. So here we have a movie, and the nice thing with movies is you can combine a series of rotations and zooms along with turning things on and off. So you can change the rendering, you can change to look at a model, you can turn those models different colors and visualize different things. So basically what these movies do is allow you to tell a story, you to present what's important for whoever's watching that video to be able to focus on. So in this case, I used a surface reconstruction for the chromosomes, and I added in a filament tracer for the spindles, and so you can see a computerized model of the original data. So that can all be incorporated into a movie. Okay. I'm not going to spend a tremendous amount of time on this, but there is some other cool features where you can pretty much tell it's an Imaris movie the minute you see these features appear. So I'm going to just add these in so I can go through them with you. For those people who are working with thick, complex tissue sections, they can be some of the hardest data sets to visualize in 3D because they just look like a block of data. And it's hard to visualize, it's hard to present. So there is a tool called the Ortho Slicer that allows you to take a single slice and image through that 3D volume. Now you can do it not only in one plane, but as many planes as you would like. And this allows you to inspect the data set, but also to present it so people can actually see the complexity without being overwhelmed with the amount of information that's there. So this one is very, very commonly used when you're trying to show someone a very three-dimensional structure and can be combined with just a regular rotation of the full 3D visualization, and then you can add in the ortho slice. We also have a clipping plane. And a clipping plane, don't worry, this little plunger tool we have here called the manipulator does not have to be in your movie, but it's here for the purpose of illustration. But it allows you to hide and show parts of the data. So you can basically drag it across from any orientation, and it will hide and show different data sets based on the orientation of that manipulator. This is one that's very commonly used when people are trying to introduce something or show a computerized model that they've made without just turning it on. They kind of slowly make it visible as they bring a clipping plane in. So this one is very, very useful and can be used to clip not only model data, but the actual image data itself. Okay. Oops, I skipped one. There's one down at the bottom here called an oblique slicer. Same idea as the ortho slicer, except you can orient that slice in whatever orientation you would like. So it's not fixed to XY or YZ or XZ. It is anywhere you want it to be. And that works really well if you have a structure that's maybe not sitting perfectly aligned with your image axis. So you can orient this ortho slice in whichever direction. Now, one last thing, and this is relatively new. We have a new feature called a reference frame, which allows you to add in your own coordinate system. So if your image is not in alignment with your image axis, you can add in your own coordinate system. And this is going to be useful for those people who want to correct for movement. This is also really useful for people who want to measure distances and actually have the origin at a point at which it's useful for making measurements. So in this case, we're looking at an egg chamber. I put the reference frame up at the top of that chamber, and you'll see that I've oriented it along the axis of that chamber. So it's something that can be very useful for making measurements because we now provide statistics not only based on distance information in the image axis, but we also do it in reference to the reference frame. Okay. So I'm going to talk a little bit. I'm not going to go into too much detail because I'm pretty sure everyone in the room has used Fiji to do some kind of image processing. So you're probably familiar with this. But I always like to point this out because not everyone realizes Imaris actually has a lot of pretty useful image processing tools. Okay, so I'm going to point out some of the more commonly used, some of the ones that I highly recommend. One is a smoothing filter. I'm sure you guys use smoothing filters in Fiji and all over the place, really. But smoothing filters, when you are bringing in 3D data, can be incredibly useful, not just for making the image look better, but for the purpose of segmentation, can make your life a lot easier. If you have a tremendous amount of pixelation and a tremendous amount of noise, it's going to take longer to generate and segment and threshold your data. Okay, because you're going to have all of this noise to contend with. So we provide smoothing filters. The one that we typically use is a Gaussian filter. It basically blurs the image based on a filter width that you can enter. The default filter width is 1 pixel in diameter, so that basically does a 1 pixel smooth throughout that 3D volume or 4D volume. So this one I highly recommend. If you can visualize noise, it's a good idea to get rid of it. Okay, so a Gaussian filter I highly recommend. Second one is one that I don't really think people use, and I use it all the time, and it makes my life so much easier. It's called the background subtraction. So I have a data set here that I got from the Allen Institute, and they basically are imaging lots of different cells and lots of different shapes and morphologies, and they gave this data set to me for the purpose of doing some other analysis, but it's very useful for highlighting this background subtraction. I'll come back to this data set though. So we have nuclei and alpha tubulin in green, nuclei in blue, alpha tubulin in green, and you'll notice that there's just so much going on, particularly in the green channel. It's hard to see where things start and end. So what a background subtraction allows you to do is enter in the smallest diameter of the structure that you're interested in, so in this case the tubules, and it will then blur the image with that value, Gaussian filter, and subtract that from the original. The end result allows you to remove background. And so what that does is it gives you more clarity. You can see the structures of interest a lot better. And that means that when you go to measure those structures, it's not going to be as difficult to pick them out. Okay? So just to make it a little easier to see, I also just have a 2D slice here so you can see this. So here's the same 2D slice. This is the original image before the background subtraction. This is after background subtraction. Okay? So for the purpose of being able to segment out these tubules, it's going to be an awful lot easier to do it with this one than this one. Now, I will warn you, both the smoothing filter and the background subtraction are destructive changes. It's changing the data in your image. So when you're using them, you need to be sure you know why you're using them and you're not just using them because someone said I should use them. They are destructive. And if you are using them, you have to use them consistently. You can't do it on one image and not on the other and compare the results from those two images. So I'm sure you're familiar with that. It goes all the way back to the way you prepare the sample, the way they acquire the sample. It's exactly the same for how you process and analyze the sample too. Okay. So background subtraction, I think I actually used it a couple of times with people in one-on-one sessions yesterday, so you'll probably see me continue to do so. Okay, another one that I use all the time is attenuation correction. So for those people who have thicker specimens, thicker samples, thicker tissue, when you image through that sample, what you typically see, the thicker it is a drop in intensity. So it's super bright at the top, and then as you image through it, it gets dimmer and dimmer. So that is signal attenuation. It's a lot to do with the complexity and the thickness of your sample. That's why clearing techniques have really helped people, because it's helped to clear out a lot of that thing, the stuff that was blocking the light. And it's also partly limitation of how far your laser can get into your sample based on the system you're using. So what we do with attenuation correction, and this is the same image we looked at before. There is a third channel, which is a membrane label. The reason it's so important to do corrections is if you don't see it, the chances are you're not going to be able to measure it. So you could have structures that are all the way through that thickness of your tissue, but if you don't see them, the chances are you're not going to segment them or threshold them or measure them. So in this case, it was a membrane label, and you'll see that for the most part, it was at the top of their sample, which happens to be the bottom here. So this is a section view showing XZ and YZ. So it only appeared in the top. In order to be able to determine the boundary of the cell, I need to see it the whole way through. So an attenuation correction allows you to measure the intensity at one end, the top, measure it at the bottom, and it does a linear correction for intensity from top to bottom. Okay? It will also amplify your background, so that's something you have to be prepared for, but there are ways that we can also optimize. So typically when I do this, I do an attenuation correction, and then I run a background subtraction afterwards. Okay. So this will really help you to pick up signal that drops off in Z. Okay, lastly, I know there's quite a few people in the room that brought tracking data to me yesterday, so I have a feeling there's a lot of live cell trackers. Photo bleaching can be a real nightmare for people who are trying to track something through a period of time. In some cases, people are not necessarily tracking intensity, they're tracking motility, they're tracking migration, they're tracking morphology changes. Those things all require you to be able to see the intensity in order to get the right measurements. So this is just showing you a zebrafish that was imaged, I think it's just the head, that was imaged for a period of time point 1, 25, 50, 75, and you'll see there's a significant drop-off in two different channels, red and green. If you were trying to measure the red objects and see how they change over time, if they're pretty much not visualized in the last time point, that's a bad sign. You're not going to be able to follow it. So we have a function called normalized time points that would allow you to take the intensity information that was in the first, intensity information from the first time point, and basically do, again, a correction so that it's applied throughout the whole time course. Again, that is potentially going to bring up background problems for you, but background subtraction can help. And one thing I do want to point out, if you are measuring intensity over time, if you're looking at calcium or pH changes and you're doing that based on intensity, this is not a good option for you because it will completely normalize any changes. Okay? So this is for people who are tracking motility, migration, size, not intensity. Okay, so I'm going to go over a couple more things before I jump into analysis. We have a new feature that is part of our blend technique. So this is probably going to be more useful for people that are currently using Imaris. So we, with the current blend technique, provide a way for you to vary the opacity of your image to make it more transparent so you can see through it. One of the issues with that is when you start varying the transparent or the opacity, you start to lose the dimmer signal. Okay, so you'll lose dimmer structures first, and the last thing to disappear is typically the bright structures. So this is some motor neurons in spinal cord. You'll see them pretty clearly at the top here, and as we vary the transparency, all that we really have left is the cell body. So you can't really see the other parts. So we have a new technique now called peak. And what peak does is allow you to put in a peak intensity and only look at a particular range of intensities, and then everything else is hidden. So the effect that that gives us is a hollow structure. Okay? So instead of varying the transparency of everything, we're choosing a particular intensity range, which gives us this ability to retain a lot of the dimmer signal but make the inside of those structures hollow, and therefore it's easier to see structures that are inside of that outer structure. So I think hopefully this helps you to see a little bit more that we can see the boundary, but the inside of these projections that previously were just gone, they're now retained and they're hollow. Okay, so highly recommend this one for anyone that's looking inside of a solid structure. It's an awesome technique. Okay. Incidentally, there's a little, so the rendering controls for blend, I will show people this afternoon, are now located in the volume settings, and there's an option to switch between step and peak down at the bottom here. Okay, so that was a little bit about visualization. So now we get to analysis. So the image that I've been using as kind of my overall workflow diagram here is these motor neurons in spinal cord. This here is a synthetic representation of the structures that we saw in the image data, also referred to as a model. Okay, so Imaris, for the purpose, not only for analysis, but for the purpose of visualization, create synthetic representations or models. Okay, now depending on the morphology of the structure you're trying to analyze, we provide different types of models. And that's what I'm going to focus on right now. Okay, Imaris has 4 different models, and the models are chosen based on what you're trying to extract from your data. It also is very dependent on the morphology of the structures you're working with. So if you are working with a neuron, then you're probably going to want to use a filament model. A filament model is for long, thin structures where you want to measure length and branching. Okay? If you are looking at vesicles, you're more likely going to be using a spot detection because for the most part they're round and you just want to identify where they are. So based on the morphology of that biological structure, you can choose the model that's most appropriate. Okay, so I'm going to go through these with you so I can explain. So the first one I'm going to go over with you is the surface. I have an 8-year-old son who lives on his Nintendo Switch, and so he checks this out all day, every day. He lives in virtual world, so he works with, or he sees surface reconstruction all the time. So a surface is basically a coating or a boundary on that is basically hollow inside. So we use this boundary to visualize the structure and how it looks. So if we look at this bunch of dappy chromosome information, this is a maximum intensity projection over on the left-hand side. What we allow you to do is set a threshold for intensity, the difference between background and signal. Once you've set that threshold, we coat the structures with a wire frame. This is an intermediate part that you don't see, but you can if you want to. And then once we put this wire frame or these triangles all the way around the structure, we then coat that with a surface. Okay? So the surface is a lot easier for the eye to visualize the three-dimensional aspect because it's now a synthetic representation. You can see light, you can see reflections, you can see shadows a lot easier. But the most important part is once you put a boundary on something, you can extract a huge amount of data. Now we know where the edge of the structure is. Now we can tell you the volume, the intensity, the diameter, the longest principal axis. We've got a lot more information. Okay? So typically people using surfaces are looking for volumetric or morphological type information. Okay. So I do want to tell you one thing about surfaces, and this is really pertaining to people who have used Imaris previously. We have just introduced a new surface model. Previously, the new model came out in summer of last year when we released Imaris 9. Previously, we were not able to create a surface on data sets or structures that were larger than about 7 to 8 gigabytes. We just ran out of memory. We had no way of handling that data. However, light sheets and stitching and new instrumentation keeps making people's data sets bigger and bigger and bigger, and so we were hitting a real bottleneck. So we use the same concept that we do for our visualization. Remember, we were talking about the rendering and the different blocks of resolutions as you zoom in and zoom out. We applied that same technology to our surface reconstruction, and now we can go all the way up to multiple terabytes in size. So this is an example from Ali Erturk. I believe he was here at Genentech, but is now University of Munich. München? Is it Munich? Maybe not. Is Munich and München the same? Yes, it is. Okay. Okay, so he's now in Germany, but this is a 72 gigabyte data set that was generated using UDisco. And he provided this data set because we wanted to test out the surface model. So what I want to show you is I'm going to show you a little movie here so you can get an idea of what we can now do. So remember I said previous to this, we could not have done anything with this data set. We would have had to do a region of interest, or we'd have to sample it down so we could make it small enough. Now we can put surfaces on huge data sets so we can actually visualize and measure whole organs, whole brains, massive reconstructions. And also, I don't know if you saw there, but I'll just run this real quick. We also not only applied a surface, oh, come on. Maybe not. Maybe that was it. You're not going to get to see it again. But we also did a reconstruction on the vasculature as well, and that's intricate, small structures. So the new model is basically giving you the ability to measure these massive structures instead of having to put them into pieces and measure them in parts. No. Oh, come on, Prezi. Okay, I think. Yes. Okay, we're back. So, sorry about that. So I do want to show you one other thing, which is the length of time it takes to generate these surfaces. So we didn't just make it an option for you to work with bigger data sets. We've actually now optimized surface creation dramatically. So what we're looking at here is another boring graph, which is showing the data set size on the y-axis. The x-axis is the time in seconds in order to generate a surface. So you'll see that there are two different color bars. The orange bar is showing Imaris 9. The gray bar is showing Imaris 8. So this is, and when I say 8, it's basically any Imaris version prior to 9 was using that same model. So with Imaris 8, we were able to go up to about 8 gigabytes in size. You'll see that it took a tremendous amount of time once you got over about a couple of gigs in size. It was a tremendous length of time. And what we've now been able to do with the surface model is cut that time down dramatically. So not only are you able to do more, you're also able to do more a lot faster. Okay, hi.
Is that time seconds per gigabyte for the image size or the size of the surface that's present?
That is for the size of the image size. So there is a certain amount of complexity that can come with a data set. As far as I'm aware, the sample was it was the same image that was downsampled. It was a embryo. It might have been a Drosophila embryo or something like that. And so they downsized it. So it was the same structure, same boundary, but just resampled for different sizes to compare.
Okay, so if you're doing something that's half terabyte, you're doing 572 times 40 seconds. It takes to render that surface.
Again, it's dependent on the structure that's in it. If it's one big solid structure, it's probably going to take less time than if it's blood vasculature and there's lots of small intricate sizes. So it will depend on the complexity. So this is just to give you an idea that it's substantially faster and able to do more. So as far as the timing, this is just an idea. It's really going to depend on the complexity of the structure you're working with. But you have a demo version upstairs, Kyle, so I would love it if you want to give me some timing information because I know you're working with massive files. That would be cool. Okay, so just to kind of take us back a little bit, I already mentioned typically surfaces would be volume, surface area, intensity values. We also provide a two-way interface so you can actually click on a measurement and see what structure that measurement pertains to and vice versa. We also do something called statistics color coding, where you can actually color code your structure based on any statistic we measure. So a lot of people use this for size differences, distance information, and also for intensity. So again, we go back to presentations. When you're looking at a 3D structure, sometimes it can be hard for people to really see those changes in size or changes in distance. So being able to apply color to that is another way to visualize that dimension. Okay, so the next one is spots. Spots is ideally suited for round-ish structures. So vesicles, nuclei, typically lower magnification nuclei, not all zoomed up. Puncta, synaptic proteins, whatever, anything that's kind of small and round, or not necessarily small, but round in shape, a spot detection will work really well for. Now, it is effectively a sphere. So what it does is it looks for a user-defined size, and we use that size to look for intensity peaks in that range. And it is effectively finding the centroid of these hotspots that are a particular size. So when we find that hotspot, we place a centroid, we place that sphere, and you can visualize where it is, but we also extract the coordinate information so you know not only how many there are, but where they are in 3D space. So this is just looking at some vesicles around a cell. So the first image is a maximum intensity projection. We then have the projection of the nucleus and then the spot detection replacing. So basically these spots are effectively just points or centroids of where those objects are located. Now, one other thing the spot detection does is it allows you to map the size of the structure you're detecting to the size of the spot. So if you have different vesicle sizes, let's say, and you want to know what the diameter of those objects are, you can actually use the spot detection to fill out the edge of that structure so you can get that diameter information also. Okay. Now, this also underwent a facelift. Actually, just a few weeks ago, 9.2 was released. And spots previously was limited whenever you hit about 1 million spots, which sounds a lot, but when you're working with massive data, it's actually not that much. But we were limited to about 1 million spots. And once you were up in that range, getting the statistics out was painful. So 9.2 was just released, and it has now been optimized so that it's faster rendering, faster creation, and faster generation of statistics as well. This is directly impacting your ability to track faster as well. Okay, so I have a movie that probably for the same reason as we saw before was not behaving in Prezi. So I just want to show you. This is a data set that we got from Genelia Research Campus. I keep wanting to call it Genelia Farms, but it's not that anymore. It's Genelia Research Campus. So this is from the Keller Lab. Kate McDowell provided a 2 terabyte Drosophila embryo data set, and the total number of spots that were tracked in this data set was 8.6 million. So there's a lot of individual structures. So I want to show you this movie that was created so you can get a sense now of what we're able to do. So throughout the whole time course, we identified each of the nuclei in every single time point, and with our tracking algorithm, we can then track each of those nuclei over time and as this is going through embryogenesis. So I just wanted to give you an idea. This, prior to Imaris 9.2, would never have been possible. So it's something that is now feasible for people with large data. Hi.
So is there like does this deal with very dense tracking? Like say, if the spots get too dense, there's a possibility of like kind of connecting the wrong spots. Is there like extra coding in there to?
Well, so that's a little bit of a different question. That's actually more to do with both spatial and temporal resolution and whether you have enough of it to be able to track those structures. And you're right. So when you see embryos growing and changing, typically more cells are formed and they become more densely packed. And depending on both the resolution at which you are acquiring your data at and that temporal resolution, that ultimately determines whether or not we can find that structure and identify it as an object, and then secondly, whether we can actually track it or not. So it's not so much whether Imaris can do it or not, it's whether or not we have the resolution in order to be able to do it. So the tracking, my rule of thumb with any tracking application is if you can visualize a cell moving from one location to another by eye, the software will also be able to track it. If you struggle, if you're looking at the data and you're like, oh, I hope Imaris can figure that out, it's probably not going to happen. So your eye is actually a really good guide. If your eye can follow it, the software will be able to. If your eye cannot, the software probably won't either. Okay. So, like I said, that's more of a limitation as to the amount of data you can acquire and the resolution. Okay, so I'm going to switch back to Prezi. So this is now available in 9.2, and I do also want to tell you that the BIDC has a demo version of 9.2 running on. I'm going to set you up on both of their workstations will be available with the new version. So if you do want to test this out, it will be available to the end of August, so you can play around.
What's the upper limit to the number of spots you can track it?
We actually haven't got there. I mean, we thought this 2 terabyte data set was a pretty good test of the spot detection, but it really ultimately depends on your computer specifications also, as to how much RAM, how much hard drive space, what your GPU, all of those things can really impact this. So we really don't know what that upper limit would be. But if anyone wants to do some benchmarking, that would be awesome. The one thing that's always tricky for us from a development standpoint is actually getting data and actually putting it through its paces. So we have a really good working relationship with multiple labs, globally that give us data, but it pretty much is just them giving us data, so it's not a broad realm of kind of data sets. So Genelia Farms is one that, I just said it again, Genelia Research Campus is one place that we do a lot of kind of collaborative work with. But if you have data and you want to push the limits, it's something that I can set you up with a demo license and you can give it a whirl. Okay. So I'm just going to bypass this one. Okay, sorry, if you're not motion sickness, if you don't have motion sickness by now, you're going to have it soon. So the last thing I'm going to mention about spots is we also have a new tracking algorithm that is specifically designed for tracking cell division. This was introduced about 2 years ago now, but it allows you not only to track migrating cells, but if they divide, we now have the option for you to track that division, so you can track division rates and generation and all of that good stuff. Okay, so that came out actually in 8.4-ish. So the BIDC currently has that tracking capability. So I wanted to point that out. And this is just a C. elegans embryo where I've used a spot detection. The little lines are showing you the trajectories, the little movement, and as those cells divide, you can see the little branch occur in the track. Also, we have a new labeling feature that allows you to label different cells in different colors, so when they divide, they retain the same color as the mother, so you can take a look at things like cell fate. So when you're looking to see where they go after they divide, it's a little easier to visualize when they're color-coded with a label. Okay. So, I'm going to keep going. Filament tracer. My favorite model, weird thing to say, but it is. Filament tracer is for filamentous structures. We typically split that into 2 different types of morphology. One is a tree-like branching structure like a neuron, astrocyte, microglia, something that has a cell body and they branch out from that cell body. This is what we refer to as an autopath technique, typically used to calculate lengths, branching hierarchy. If anyone is looking at neurons and interested in dendritic spines, it also detects spines as well as spine morphology. So this one is typically what we refer to as the neuroscience algorithm. We also have another algorithm that is for anything that's not a tree-like branching structure. So it could be a looping structure, it could be a network. My example here is some blood vasculature. This technique is called threshold. It effectively segments the data and thins it or skeletonizes to 1 voxel in diameter. The benefit is you can get all of the length and branching and diameter information that you need from that structure, but you don't need to have a start position like the autopath technique requires. So anything that does not have a defined start location would be a threshold technique application. So this, incidentally, this data set up here, this is just a small portion of it, was a 20 gig expansion microscopy expansion data set. And each of these neurons were actually traced independently. We've also optimized our semi-automatic tracing tools to work with big data. So that's something that I'll be showing at the very last session today, some of the new tools for that. Okay, I think I have a little movie here, or maybe I don't. Oh, yes, I do. Okay, I'm going to be showing you, if time permits, I'm going to be working with this data set a little bit later on. This actually has a second channel with microglia in it, so I want to show you how we can look at interactions between different structures like blood vessels and microglia, or blood vessels and pericytes, that kind of thing. But just wanted to show you, this is the filament trace of the blood vasculature in there. Okay. As far as statistics, I already mentioned length, diameter, branching hierarchy. We also do shoal analysis for those people who are interested in complexity of branching, and then spine statistics. And lastly, any model that I'm talking to you about this morning, are trackable, meaning that if you're interested in tracking a filamentous structure over time, we can generate length and branching changes and things like that over time for each of those structures. So there seems to be a lot of people interested in tracking microglia, morphology and projections, and so this is something that can be done using the filament model and the tracking tool together. Okay, I will zoom to the next one. Okay, and last model, also our newest model, meaning it was developed the most recently, is called cell, and the little icon for the cell model should hopefully explain what it is. So instead of looking at one structure and segmenting that one structure, it allows you to segment multiple compartments within a boundary of a structure. So the icon is representing a cell, the blue is the nucleus, the red is the little vesicles. So the benefit of something like the cell model is you can identify numbers and amounts of structures within another structure, okay? And you can get those statistics on an object by object or cell by cell basis, okay? So I have two examples here. One thing that's unique about the cell model is it has a membrane detection algorithm, okay? So it means if you are using a membrane label to define the boundary of your cell, this is the only place where you can actually get morphology or boundary information using a membrane label. Okay, so this is just the same data set I showed a little earlier from the Allen Institute. These are actually, it was just one complete cell in the center of that data set. So the cell model allows you to segment out using that membrane, so you can actually then get statistics. And in this case, I think this was actually mitochondria they were looking at inside of this cell. So not only can you find the boundary of the cell, you can then get nuclear volume, amount of mitochondria, number of vesicles, overall intensities, whatever it is that you might need from that. And then down here we also have a cytoplasmic detection, which is like a surface. So you're just basically looking for a filled structure, you're putting a boundary around it, and then you're finding other structures within that boundary. Okay. You get to choose what compartments that you are going to be measuring. So the compartments are cells, nuclei, and vesicles. That is what they're named, but they do not have to be used to measure those compartments. So we have a lot of people looking at fish or doing fish analysis where they would actually use the nuclei as the cell, the boundary of interest, and then within there they would count the puncta with a vesicle. Okay, so I wish it was not labeled with a name, although they're trying to make it a little bit more biologically smart, so they're giving compartments names. It would be actually better if it was just compartment 1 and you got to name it. That's coming in the future, but for right now I would just want you to know that these compartments can be used for whatever structure you want to use it with. Okay. So once you've identified the compartments of interest, it will then split all of your cells. Typically, when you are working with multiple cells, they are very close to one another, so we have multiple splitting algorithms. And then the statistics, as I said, are given out or produced on a cell-by-cell basis. So if you're looking at counts or nuclear volume or intensities, it's all done on a cell-by-cell basis. And then lastly, distance. I'm going to show you distance in the introductory and the advanced tutorial sessions today because it's really, really important for a lot of people. They want to know where things are in relation to something else. So the cell model automatically does that for you. So if you have multiple objects inside of another object, we automatically calculate where it is in reference to all of the other compartments. So the color coding on these vesicles here is actually representing its distance to the cell membrane. So that statistics color coding helps you to see those distance measurements. Okay, now it is 11:10, so I'm probably going to skip a little bit of my last section so I can show you something in Imaris. But the last part is once you have generated models, you will have generated thousands and thousands of different statistics. There'll be a lot of information that you have to kind of go through and figure out what to do with. So I wanted to show you an example of where our data interpretation can be helpful for you to try and decipher what's going on. So I actually saw somebody working with dendritic cells and T cells yesterday, and this happens to be an example that I'm going to use right now. So we have an image here. The red is the dendritic cells, the green are T cells, and you'll see that I generated a surface model for the T cell and a surface model for the dendritic cells. I'm going to play, and then I'm going to play a movie. So you can see that it is a 4D data set and that there is a lot going on. The T cells, the dendritic cells are all moving. I'm adding in the surface model so you can see that they're representing each of those different cells. And then I'm also showing you the tracking information for the T cells. So voila. Okay, there's your data. It's a lot. There's a lot of information in there. And so what I want to try and show you is how we can kind of get some information out. So I'm going to do that by introducing you to a module that is available at the BIDC. It came out quite a while ago, but people don't realize it's there. It's called Vantage, and it's a way to statistically plot using your model information. Okay, so with this Vantage module, we provide 4 different plot types. There's one called XYZ, there's one called gallery, there's one called time, and then we also provide a scatter plot too. I'm going to give you examples of what you can do with the plots that may help decipher or to, get some information out of this data set. So the first one I'm going to look at is one called the XYZ. So what this does is it leaves the data in its original location. So XYZ meaning it's in its original XYZ coordinate location. And what you can do in this view is statistically color code. So we talked about it a little bit already. So color can be used to represent any statistic we calculate. So in this case we have volume. So smaller structures will be cooler, bigger structures will be warmer. And then we also have the option of adjusting their scale based on those statistics too. Not super interesting for dendritic cells, but really helpful for this one up here. So this is another movie, and you'll see that the color coding has been applied to the T cells here. So what I'm going to do is I'm going to play the movie, and I'm going to see if you can recognize what the color is representing. Anyone realize? Exactly. So the purple is representing, the color is representing distance. Okay, so things that are purple are 0. That means they're actually interacting with those dendritic cells. And the warmer the color, the further away they are. So just by applying color allows you to see and visualize the interaction between those structures. Now, obviously, we have all the data, but it's a way for you to see that. Okay, the second is a gallery view. And this one is really useful if you are looking at cell morphologies. So if you are looking at changes in morphology, if you're looking at comparing cell morphology after some kind of treatment, anything that involves shape, different shapes. The gallery view is really cool because it allows you to take all of the structures out of your data set and put them into a grid. And you can sort them based on any statistic you want. It could be volume, it could be intensity, it could be the longest principal axis. So in the case of the dendritic cells here, I've basically sorted these based on their longest principal axis. Okay, the stretch for the dendritic cell. So that can all be done, so you're basically seeing them all in a grid at the same time, and it can be really useful. A lot of people, I keep saying microglia, it just has to be, it's a hot topic right now, but the microglia community really like this feature because they can see everything at the same time and organize that data. Okay, not so useful with the T cells because they're all kind of the same morphology. However, I just wanted to give you the comparison that gallery is really useful for morphology. Also, a lot of people use gallery when they're looking at blood vasculature and they're looking at the different segments of blood vessels. They can look at different thicknesses and sort that based on diameter too. So it can be really useful. Also, I'll point this out. We have a statistic called bounding box. For Imaris users in the room, if you haven't used it yet, you should use it. It's a cool feature. It's a cool statistic, sorry. All it does is give you two options. You can place a box around your structure, and it will calculate the longest principal axis as well as the shortest principal axis, and then the second longest principal axis. So for people who are looking at changes in morphology over time, this statistic can be really useful for that purpose. So I wanted to point it out. It's not something that's automatically checked on, so you'd have to go in and turn it on in order to use it. But that is a useful statistic. Okay. We also have a time plot. Each one of the points on the plot is the surface reconstruction of that object. So if you are looking at changes in the structure over time, this is not only giving you, this is actually stretch basically versus time, but you can actually see the change in the morphology over time because you're using, instead of a point, you're using the object itself. And then also we give you box plots, so you can look at min, max, mean, first and third quartiles as things are changing and moving over time. Okay, I zoom up on there. Okay, and this is the last one. The scatter plot is completely up to you. We give you the ability to choose whatever statistic on the X, the Y, and the Z axis, and you can then do your own comparisons. So I'm giving you a couple of examples here, one for the dendritic, one for the T cells. So for the dendritic cells, because we're probably mainly interested in what's happening to its morphology as the T cells are interacting, what I wanted to show you is something that looks a little bizarre, but it's really cool. It's basically the cells plotted over time and color-coded based on time. So what you're looking at here is the cell in the first time point. The purple is representing your first time point. And then as you go through Z, that is the morphology changes. So if there is more projections coming out of a cell, or if there is a change in size, you can apply this color coding and be able to see all of your time points simultaneously to give you that information. So wanted to show that one. Okay, second one. You may have seen these plots before. You can generate them in Imaris, but this is also a feature in Vantage. This is a displacement plot. So you can basically look at the tracks of all of the cells in your data set, and you can have them all start from the same origin. So basically have them all start from 0, 0, 0, and look to see where they go from that point. So are they all migrating in one direction? Are they randomly moving? Are they moving far? Are they moving nowhere? So I split these into two different groups. The cyan is representing those that are interacting with the dendritic cells, and the magenta is not. So obviously the ones that are not interacting are going further. So this plot is just a really cool way to display what's actually happening. And the big difference here with what you would see with a typical displacement plot is that you can actually turn all the time points on at the same time, so you actually see morphology changes along with that as well. Okay? Okay, I'm going to finish there. There is more, but I will finish right there because I want to jump into Imaris and just show you a couple of examples. And for this, I'm going to sit down. But while I'm switching, does anyone have any questions at the moment? No. Yes?
What are the options for batching? Some data processing options?
Okay, that's a great question. Okay, so I should probably turn the recording off because I'm going to give you some information that I probably shouldn't be giving you. Currently, Imaris does not batch process image processing functions. So smoothing, background subtraction, attenuation correction, those cannot currently be batched. However, creating the models, the surfaces, the spots, the filaments, the cells, all of that can be batch processed, not the image processing component. The batch module is basically designed for you to put a bunch of data into a particular group, and then you can apply the same parameters to that group. Image processing functions will be available in Imaris 9.3, which is coming out in the fall. So that means you can combine those image processing functions that are currently available in Imaris with those creation parameter routines. So the reason I shouldn't have said that is because I'm not meant to release what's coming, but it is pretty close. We're like a couple months away. It's not far. So it's actually due to come out in November. So for those of you that are interested in batch processing and getting all the image processing done, that would be something that happens in November. Okay. So I'm going to jump into Imaris, which I believe I have running in the background. Okay. Okay, so I'm using this vasculature data set that I used a little bit earlier. It's a 2-photon image from a mouse brain. The green are the microglia, and the red is the blood vessel. And what I wanted to go through with you is a couple of features that you may or may not be familiar with that can actually be really useful. And they can be used across the board in lots of different experiments and lots of different data sets. So first thing I want to show you is we have obviously the ability to model the different components within the data set. So in this case, I just turned on my vasculature surface. And I'm going to turn the volume object off so that we can see the blood vasculature on its own. So we have a reconstruction of where the blood vessels are. Now, one thing that we can do with surfaces is something called masking. So I'm going to show you a couple of things, and it might be easier to look in the slice view so you can see this a little bit better. It's pretty dim on the projector, so I'll kind of max this out and bring this down. Okay, so this is the original data. So if I was to zoom up and down the side here, you can see the blood vessels and the microglia as I go through the data set. You'll also notice there's a tremendous amount of background. Okay, now a background subtraction would definitely help, but there are actually some structures in here that's not vasculature that is still fluorescing, and even a background subtraction would not necessarily remove it. So I want to show you another feature of a surface that can be very useful. So if I go back to the 3D view and we take a look, and I'll turn this down again, and take a look at the surface, as I've mentioned already, it's effectively a boundary. It's a 3D boundary. And what we can do with that boundary is we can tell it to do different things with the voxels that are inside of the boundary versus outside of that boundary. So if we click on that surface object, there is an edit button, and in that edit button there is a feature called mask all. Now what the mask function does is allow you to treat data inside and outside the surface however you would like. So in the case of blood vasculature, there's a lot of other stuff that's fluorescing that I don't really want to see and I don't want to measure. So I'm going to choose the Psi3 channel, the blood vessel channel, and you'll see there's an option here that says set voxels outside of surface to a particular value. Currently by default it sets it to 0. And if I click OK, it will generate a new image. And that new image, okay, so let me turn off the surface so we can see this. So here we have the original as well as the new channel. So if I turn off the original channel, you'll see that the new channel gets rid of all of that background. Now, the reason I'm showing this is when it comes to segmentation, when you are trying to trace something or track something, if there's other stuff in the field of view, it can cause tremendous amounts of problem, but it can also take longer to track. So you have to find them all and filter them somehow to get them out of your data set. So this tool is a way for you to easily do that. Okay? The other tool, or the other feature I wanted to show you, is regarding distance information. So you'll see that I have not only blood vessels, but I have microglia. Now, I know that they didn't look that crazy before I had the intensity turned down on these microglia, but let me show you what these guys look like. So they happen to be extremely bushy. In this particular data set, they happen to be extremely bushy. There's a lot going on and there's projections everywhere. So even though the display wasn't showing them, I used the surface segmentation to pull out that very fine, dim projection information. And so what we have here is a surface model that represents those microglia. Okay? Now, one thing I want to show you is if we wanted to see all of those microglia that were within or that were interacting with the blood vessels, we can do something called a distance transformation. And a distance transform is the way for you to calculate a 3D distance map so you can measure structures in reference to something else. So if I have my blood vessel as my reference, if I go into this little gear icon at the end, it's called our Tools tab, there's an option in there called Distance Transformation. And that will create a new channel that stores distance as an intensity. Now, I've actually already created this, so I'm going to go into the slice view so you can see what this looks like. Okay. I'm also going to change the color of it so it's easier to see. I'm going to change it to a heat map. Okay. Okay. So do you see this bizarre halo around the image or around the blood vessels? This is a distance map. So I'm basically color coding the distances so I can see things that are close or far away. And so you can see these little bits of blood vessels throughout the image, and then you see this kind of change in intensity around the edges of them. This is a 3D map, so as you move through the data set, you'll notice that depending on where that the vessels are, the distance map is going to change. Now, I'm going to use this distance map to now identify all of the microglia that are touching the blood vessels. And I can do that because if a microglia is touching a blood vessel, the distance of that structure will have a distance of 0. Okay? So I'm going to go back to the 3D view, and I'm going to turn on the microglia. I'll turn the volume off for a second. So we have our two different channels. I'm now going to go into the microglia channel and go into the statistics. And in this detailed view, there is an option that allows you to pull out that distance information. It's an intensity value that pertains to the channel where the distance is located. So in this case, it's channel 4. I'm going to click on that, and these values in here represent the distance in microns. So if I want to see that object, I can click. It will highlight the object, and obviously there's a lot going on, so I'm also going to center to selection, and you'll see there's a little microglia in here highlighted. So I'm going to sort these and show you that you can come in now and measure or select all of those that are within 0 microns of the blood vessel. Now, we can do this in the Statistics tab. We can also do this in the Filter tab, which is designed to allow you to filter any statistic you want. So in this case, I'll go in and do the min for channel 4. Sorry. I forgot my mouse today, so I'm scrolling around. Hang on. Here we go. And then in here is where we can basically set up what our min and max for that threshold would be. And now everything that is highlighted in yellow are things that are interacting with the blood vessels. We can then duplicate into a new object, so we can actually get counts of all of the microglia in total versus all of those that are interacting with the blood vessels. So this is something that doesn't have to be for these particular components. It could be for multiple different experiments. And this is a fixed tissue, this can also be done in 4D or in 2D over time. So if it's a tracking component or if it's a fixed tissue, distance information can be really useful. So in this case, I'm also just going to show you if Vantage will agree with me. We'll see. Vantage will also allow you to take all of those, not the spots, I want the surfaces. There we go. So I'm going to take all of the microglia and I'm going to sort them into a gallery. And this is really what I wanted to show you. So each one of these objects is representing a single microglia. And I'm probably going to take a look right now at the longest principal axis is the way I want to sort them. And I'm also going to color code them here based on, let's say, their volume. Okay, so this is now the Vantage view of all of those cells that were picked out to be touching the vasculature. So now you have a way to basically categorize them based on their interaction and then take a look at their morphology differences between those two different groups too. So I just wanted to give you a little bit of a kind of visualization as to how we do some of those things in Imaris. Incidentally, all of the plots that you create, you can take snapshots and export them out. Also, all of the statistics that we use in order to generate the plots are down below and can be exported out to Excel. All of the statistics also in our 3D view can be exported out to Excel as well if you prefer to use something else for plotting. Anyway, hopefully that gives you a little taster of what Imaris can do. If you guys have any questions, definitely direct yourself to the BITC, or if you have anything for me specifically, I'm the one that's been bombarding you with emails, so I know you have my email address. So if anyone has any questions, please feel free to ask around. And for those of you that I'm going to see this afternoon, if you're in my last session, I might not be able to form sentences, so I hope you come prepared. But we're going to be setting up in the computer lab, which I believe is across the hall. So this is going to be more of a session where you get to follow along and see the workflows as to how we create structures and how we can pull out the statistics. But if anyone has any questions right now, feel free to ask. Otherwise, you are free to go. Thank you.
Thank you, Andrea. If everyone can make sure that they signed in on the sign sheet before they leave, that would be awesome. And